Hi everyone, thanks for being here today. My name is Kristin, I'm from Griffith University on the Gold Coast. I'm going to be talking to you today about how fishery food webs vary among different seagrass types. A couple of my colleagues are here, Rod, not sure where he's sitting at the moment, Rod up the back there, and Abby. And we're working as part of a broader collaborative project with Griffith University, the Gold Coast, and James Cook University in Cairns. So I live in a pretty awesome part of the world in order to study coastal ecosystems such as seagrasses. That's me in the orange dot there, just outside the Great Barrier Reef. We had a fantastic introduction from Alana this morning about how much seagrass is actually in the Great Barrier Reef, so I might skip those numbers, but just to remind you, there is a lot of seagrass in the Great Barrier Reef. The work that I'm doing in my PhD is focused mainly around the support that seagrasses provide for the ecosystem service of fisheries production. So to give you a bit of an idea of what the Queensland coast actually looks like and what these impacts are, what the impacts are on our seagrass systems, I've got some local photos here. Top left is the Gold Coast, so it's a bit outside the Great Barrier Reef, but it's still an example of the widespread urbanisation that we're seeing. We've got land reclamation, this is in Townsville, and then we have port and coastal and port infrastructure that's, this is located in Gladstone, just in the southern end of the Great Barrier Reef. So this is what's facing our seagrass systems today. We know that this is having a big impact, but one thing we don't know is whether our seagrasses are all going to respond in the same way. So our seagrasses come in all shapes and sizes and different levels of complexity. And as Lena said earlier, even within the same seagrass species, she stole my line, you can have tall versions and short versions, even within the same seagrass species. To some of us, some of these species might look the same, but that doesn't necessarily mean it looks the same to the fish. So we wanted to know, is there something about the structural complexity across all of these different seagrass meadows that is related in some way to fishery food webs? So we already know quite a bit about how important seagrasses are for fisheries. First of all, we know that there are important links with adjacent habitats. In tropical systems in particular, these important habitats are mangroves, coral reefs, links to continental shelf fisheries, and then of course through detrital transport to our deep offshore fisheries. We also know from research that different types of seagrass species can actually suit different groups of animals. So for example, fish prefer a different species to potentially what crustaceans would go for, and this has been shown through isotope analysis. We also know that there are a range of drivers of change to our fishery food webs. We have top-down processes through higher order consumers that are controlling those fishery food webs. We have, in some situations, shown that eutrophication can impact the structure of food webs. We also know that at seascape variability, at the broader scale, this can also influence food webs. And finally, life history traits and meadow form have also been shown to have differing impacts on the way food webs might be constructed. There is also evidence, however, that there are relationships between animals at the individual meadow structure level. And so we hypothesised that at that individual meadow structure level, there potentially would be links with bottom-up control processes from the structure of the meadow to the way that those food webs are constructed. For our experimental design, we picked 10 different seagrass meadows that spanned around 900 kilometres of the Queensland coast within the Great Barrier Reef World Heritage Area. We picked meadows that were all exposed to the same sorts of fishing pressure. They all have similar eutrophication impacts and distances to estuaries. And so we spread those meadows across the coastline. The 10 meadows that we selected ended up falling across 5 different species into 4 different genera. At each of these 10 meadows, we took subsamples so we could measure complexity variables. We quantitatively sampled for animals that fell into 4 different trophic groups, so predators, omnivores, herbivores and detritivores. We then sampled basal plant sources so that we could construct isotopic food webs, so seagrasses, and then we also collected epiphytes off the seagrass and benthic microalgae. We ran the usual carbon and nitrogen, which is pretty typical in all isotopic food web studies. But we also use sulfur. So we analysed all of our animals and all of our plant sources for sulfur. And for those of you in the field, you'll know that sulfur has come a phenomenally long way, and it's proving itself to be an important splitter when it comes to determining food sources in these food webs. So by way of example, when we run our isotopic mixing models, often you'll have a similar carbon and nitrogen isotope signature, and it's very difficult for the model to give you a definitive answer on that food web construct. When we include sulfur, however, it gives us that split and can give us a more definite answer. And that's because seagrass takes its sulfur from the soil, from the sediment, and epiphytic algae and benthic microalgae take their sulfur from the water column. So I've used sulfur across all of my studies, and I will do for the rest of my PhD. So the habitat variables we measured were fourfold. We measured above ground biomass, the canopy height, we measured leaf area index, and the shoot density. So these formed the basis of our analysis for structural complexity at the meadow level. We also looked at a number of other variables. We had genus, species, depth. All of our meadows were considered shallow, but within a few metres of each other, so we had a little bit of variability there. And then we categorised all our meadows into two different classes of leaf form. So we had the Halophila, two species of Halophila in one category, and then, so they were the round, flat leaves, and then the other category was all the long, strappy leaves. So what we did with these other variables was just run a series of ANOVAs and ANCOVAs to see if there was any relationship with our complexity variables and the food web metrics that we measured. Nothing turned out to be significant, so we proceeded using the complexity variables on their own. We had three groups of food web variables that we measured. So community composition, we looked at species richness and diversity. For biomass, we looked at the total biomass of the whole meadow, and then the biomass of predators. So that's a little bit, I was meant to be a bit more centred than that. We had predators, omnivores, and then, it hasn't come up, but the bottom, the green there, we actually pooled the herbivores and detritivores into the one trophic group. We then measured the seagrass contribution to the diet of each of these different trophic groups. So key findings overall, and then I'll go through what those findings were. Species richness and diversity was pretty much the same across most of the meadows. There wasn't any significant variation there. For the herbivores and detritivores, there wasn't much variation there either. However, with the predators and omnivores, we found a relationship with biomass, and we also found a relationship with seagrass contribution, which I'll go through. So basically for predator biomass, we found that with increased canopy height, we had an increased number of predators, increased biomass of predators. So because of this relationship, we wanted to know, was it the number of predators that was increasing? Was it the number of different species, or was it just overall biomass? There was just more of the same sorts of species. So we had a look at the major players that might have been contributing to this, and there were three. So tropical snappers, emperors, so members of the emperor family, and flatheads, and these are all key fishery species in the tropical region. So they were having a major impact on how that biomass was increasing with canopy height. Omnivores showed the same relationship. So canopy height played an important role in the proportion of omnivores within my biomass food webs. So what that looks like is at the high end of the scale with a high canopy height, we had a high proportion of omnivores in that middle trophic group. But then on the other end of the scale, we had a very, very tiny proportion of omnivores in that group. When it came to the contribution of seagrass to the diet of these animal groups, we found three of our complexity variables showed a significant relationship. Now, because of the multiple collinearity between these three variables, we ran them through a principal components analysis. The PCA provides you with a single variable, a single value for each meadow, and that explained 97% of the variation, so we went ahead and ran our analysis using the PC1 value representative of our complexity metric. So we found that as our PC1 value increased, or as the complexity of each of our meadows increased, we saw a significant increase in the contribution of seagrass to the diet of both predators and omnivores. So what that looks like is that when we have low complexity, using with those three variables in there, there was a high contribution of algae to the proportion to the diet of both groups of animals. On the other end of the scale, with an increase in complexity, there was a high proportion of seagrass contributing to the diet of those animals. So in summary, we found canopy height was the most important thing for biomass of both predators and omnivores. When it came to seagrass contribution, it was the complexity of the whole meadow. As that increased, so did the importance of seagrass to the meadows. So this is potentially an indicator for us to know which seagrasses are potentially important for fisheries, specifically to the Great Barrier Reef World Heritage Area. And that's me. Thank you for listening.
We have time for questions.
Good job, Kristin. Just a quick question. Did you just take samples for isotope analysis from like the seagrasses and the algae? Did you look at anything else?
Seagrass, epiphytes and MPB, and we had those, took the isotopes for all the animals as well. So we sampled and took a select number of predators, omnivores, herbivores and detritivores. So in each category we had a broad span of species and analysed those. Yeah.
But could there have been any other contributing sources? So that like in those areas where you sampled, like was there mangroves or anything else around where you sampled the seagrass?
No, so the meadows we picked they weren't near other habitats. They were right in the middle. We went right to the middle of a particular meadow, far from mangroves or salt marsh. They were intertidal to subtidal, but even the intertidal were pure seagrass meadows, and we sampled within those meadows. So it was small scale sampling. So, yep.
Cool.
Good afternoon. I'm wondering what kind of mixing model you're using, because the contribution from various sources might vary depending on the model that you're using. Thank you.
Yep, using mixed IR models. Yeah.
Sure.
Yeah.
Have you tried other models?
I did. I actually used isoera as well, and I found I got similar results using isoera. But Phillips, do you know Don Phillips?
Yeah.
Isoera? Yeah. So I did actually, I ran a couple of them, not every single one, but some of the most prominent results, I ran them with isoera and they gave me very, very similar numbers.
Okay, thank you.